To introduce Dr. Ashutosh Sharma, a highly accomplished academic and researcher in the field of agriculture science, specializing in plant pathology. And Dr. Sharma has a strong educational foundation and extensive research exposure following the UGC 2009 regulations. His doctoral thesis focused on the of leaf spot disease in Vidania somnifera ashwagandha, employing morphological, biochemical, and molecular approaches. He has 5 years of research experience in plant pathology and plant biotechnology. Key areas of his expertise and contribution include he also worked on PCR-based detection of leaf spot disease in Vidania somnifera. He is a prolific contributor to the field with 35 research papers in journals, 36 book chapters, a Google Scholar in H-index of 13, and 595 total citations. He has also submitted 3 microbial cultures to MTCC and 3 GenBank nucleotide sequence. Dr. Sharma has received multiple honors recognizing his research contributions: Senior Scientist Award 2022, Scientist of the Year Award in Plant Pathology 2022, Young Investigator Award 2019, Young Scientist Award 2011. He was qualified for CSIR JRAF 2007 and ICR NET 2010. He has completed numerous MOOCs, including a postgraduate diploma in Agriculture Extension Management from Manage Hyderabad. Dr. Sharma has extensive experience in both research and teaching. Currently, he is Associate Professor at DAV University since 2022. His courses cover a range of specialized subjects including phytonematology, mycology, advanced virology, integrated pest management and disease management, and plant pathology for UG, PG, and PhD students. He is an inventor on 3 recent Indian design patents related to agriculture technology, including an IoT-based biofertilizer and biocontrol device and a plant health monitoring device. He has served as a PI or co-PI on 4 completed and 2 ongoing research projects funded by agencies like SCRV and CSIR. Dr. Ashutosh Sharma brings a wealth of expertise in plant health management and agriculture innovations. Please join me in giving a warm welcome to Dr. Ashutosh Sharma. Thank you.
Thank you, Gurpreet, for introduction. Am I audible to everyone?
Yes, sir, you are audible.
Yes, sir.
So I'm sharing my presentation. Is it visible now?
Yes, sir.
So today I will talk about mycology, plant pathology, and gradually I will shift on how plant pathology can affect the quality of herbal medicine. So first of all, to start with, I will discuss mycology. Mycology is study of fungi, a systematic study of fungi. There are more than 120,000 species of fungi which have been reported till date, and estimated number is much more. Actually, reported are less, but many more species are expected to be there. Some familiar names in fungi, some types of fungi which we know, they are mushrooms, they are yeast, they are other molds. They have some beneficial roles to play, and they have some harmful roles. They actually acquire their food by absorbing, by degrading macromolecules into smaller molecules and then absorbing them. Actually, their body is made up of fine thread-like filaments known as fungal hyphae. These hyphae could be branched also. They contain numerous cells. Cells are arranged in the fashion, in the linear fashion, and the thread-like structures, which are called as hyphae, they have all the cell organelles in them. They can be monokaryotic, dikaryotic, and they can be cynocytic, depending on the type of fungi. Karyotic, they are not like prokaryotes, not like bacteria, which do not have a well-defined nuclei. They are heterotropic. They cannot synthesize their own food. like plants can synthesize their own food, plants can photosynthesize, not photosynthesize. It has to take its food from other places, from dead and decaying material or from the living host, right? So it is also similar to plants in a way that it have cell wall. Animal cells don't have cell wall, plant cells have cell wall. Fungal cell wall is usually made up of chitin, but few primitive fungi belonging to oomycetes, they have a cell wall made up of cellulose. Some fungi, they are very good for human nutrition, like mushrooms, whereas some other are highly toxic. They can kill humans. As far as their structure is concerned, fungal colonies can be microscopic, very minute in size, which we cannot observe with the naked eye. We can see them only with the help of microscope, whereas some other fungi like mushrooms, their body, fruiting bodies, we can see they're macroscopic. Fungi have different roles to play. It is a scavenger also. Decaying material. It is a symbiont. When it lives alongside algae, they live hand in hand. Algae benefits fungi and fungi benefits algae. So such a symbiotic relationship is called as lichen. They are used as a foodstuff, like mushrooms are there, which we can eat, like button mushroom, oyster mushroom, milky mushroom, milky mushroom, gucci mushroom. They also act as plant pathogens. They can do disease in plants. Like here in the bottom image, apple is affected by a fungal pathogen. There is apple scab on these apples. As far as reproduction is in fungi is concerned, fungi can reproduce both by asexual as well as sexual means. If a hypha is fragmented into smaller parts, so the smaller fragment, when it is placed on the other nutrient environment, maybe on a different petri plate, it can grow. So this is called as fragmentation. Yeast cells, on the other hand, yeast which is used in baking industry, brewing industry, yeast cells, they grow by budding. And the fission yeast, Schizosaccharomyces genus is there. It divides like bacterial cells by fission. So in asexual reproduction, lot of spores of fungi, they are formed. Different types of spores. Spores are actually small structures which are, when spread, they are blown away, maybe through wind or through their like flagella of zoospores, or by any other mean. When they are blown away, they are taken to different places. They can give rise to new colonies. There can be arthrospores which can pinch off from a growing tip of a hypha. There are chlamydospores which are very thick-walled. They can resist the conditions like extreme environments, extreme heat, extreme cold, extreme weather conditions. These spores can survive. There can be small conidiospores also, which are spread by wind, and they fall on a new substratum. They find new nutrition and they can grow. When there is sexual reproduction in fungi, when we talk about sexual reproduction, there is involvement of two gametes, one from one type of colony, maybe you can say male, or the other you can say female, or if they are not differentiated like male or female, you can say of plus mating type and minus mating type, of two different mating types. So they can fuse together and form a new zygote, which can give rise to new hyphae. There are different means. Isogamy, when the gametes are of same size, and isogamy when one is bigger, one is smaller. Oogamy, when the egg like structure is there on side, and a motile sperm or motile cells gamete comes and fuses with it. There are spermatia formation, like in wheat rust. So one spore can come and it can reach receptive hypha, then fuse with it. There are formation of gametangia on side-by-side hyphae. They can fuse to form a zygospore in zygomycetes. these gametangia. In oomycetes, a female part is oogonium and thridium is male part. They lie side by side or one above the other, and they can exchange their material. And thridium can give its material to oogonium. Then zygote can be formed. So this way, two different gametes can be there. They are there rather in sexual reproduction, and when they fuse, there is sexual reproduction. This leads to formation of new combinations, because when they fuse, there is diplotization, and after diplotization there is haplotization, and in between there is meiosis, in which there is crossing over. New combinations of genes are formed. So it is good when there is sexual reproduction in fungi from fungal point of view, because it leads to formation of more variation with the population of fungi. Actually, when we talk about fungal, we confuse that what are they? They are usually the structures which are used by fungi to spread. Like there are seeds in plants. When the seeds are spread to new places, they can germinate, they can give rise to new plants. But there's a big difference. The difference is seeds have embryo in them. The embryo grows. Embryo has a well-defined like root part and shoot part, radicle and plumule, and there is a polar small miniature plant within the seeds, which is called as embryo. Whereas in fungal spores or simply spores, they can be one cell, more than one cells can be there in a spore, but they're vegetative cells which can give rise to new hyphae, new colonies. So this is the major difference between spore and a seed. When the spores are formed in fungi, there are different mechanisms to spread the spores. Like in puffball, forcefully spores are released. In Pilobolus, there's a fungus which grows on cow dung. In Pilobolus, there's a black cap-like structure. This photograph on the right-hand side, top Pilobolus. This Pilobolus, what it can do, it forcefully discharges its contents within a fraction of a second. This is a very beautiful mechanism. You can find the videos on maybe YouTube where Pilobolus is throwing out its hat-like structure and exploding its spores out. There are stink horns on which flies can come. It gives foul smell, and these flies can come with their body parts. Spores are attached, and wherever flies go, they carry the spores. Likewise, earth stars are there which burst open, and this way they spread their spores. If we talk about history of mycology, it is like prehistoric times. From prehistoric times, the mycology was developed as a science. Amanita mushroom, which is a poisonous mushroom, the wife of Roman Emperor Claudius Caesar used the soup to kill Claudius. Sir E.J. Butler was first imperial mycology, systematic study of mycology in India. So this way, in India, mycology progressed. The term mycology was coined by M.J. Berkeley. And there are many historic events. P.A. Micheley is generally regarded as father of mycology. He wrote a classical book called as Nova Planetarum Genera. And then everybody knows a person called Alexander Fleming. He was the main person behind the discovery of penicillin. It's an antibiotic which saved the life of millions during the Second World War. I suggest everybody to go through this beautiful article, History of Mycology from India, whosoever is interested in reading history of mycology. There is small glimpse, some events from the history. So there's a screenshot. Now, fungi has both harmful and beneficial roles, as I told you earlier also. So first we will discuss its harmful roles. It can spoil our foodstuffs, our fruits, our pickles, bread. It can cause diseases in humans, skin diseases, Then there are diseases on plants, which we are going to discuss in next part of the presentation. And it can spoil leather bags, handbags, leather shoes, and even leather jackets. If we talk about moles, fungus gives us food. Fungus gives us medicines. Fungus gives us antibiotics. Fungus gives us, they give us the dyes, different type of coloring molecules which are used to dye our clothes. So briefly classifying fungi, the true fungi, Eumycota, has been distributed into Mastigomycotina, Zygomycotina, Ascomycotina, Basidiomycotina, and Deuteriomycotina. This is as per Ainsworth. There are many classifications of fungi. Ainsworth, yet it is an old system of classification, but it is beautifully explained. And today also, some mycologists, they follow Ainsworth's classification, although many new classification systems have come. Myxomycota, unlike the true fungi, Eumycota, they contain slime molds. So if we talk about Eumycota, true fungi, there are Mastigomycotina, which contains oomycetes also. The sexual spore sometimes in oomycetes is oospore. In Zygomycotina, sexual spore is zygospore. In Ascomycotina, sexual spore is ascospore. In Basidiomycotina, it is basidiospore. So basidiospore for Basidiomycotina, ascospore for Ascomycotina, zygospore for Zygomycotina, oospore for oomycetes. Deuteriomycotina contains the fungi in which no sexual reproduction is reported till date as per Ainsworth's classification. So fungi, beyond its roles as a harmful agent for humans or beneficial agent for humans, they can cause diseases in plants. And besides diseases, they sometimes do very good to plants also, besides causing diseases in them. So they can form mycorrhiza with higher plants, in which the hyphae of fungi and the roots of higher plants, they work together. The fungi gives nutrients to water and some vital nutrients to plant, and plant makes food, give it to fungi. There are some endophytes which grow within the plant but without harming plant. So endophytes also sometimes give disease resistance capability to plants, and many of these fungi, they are causal agent for plant diseases. So in the second part of the presentation, we'll be discussing what is plant pathology. Plant pathology is the study of diseases in plants, sufferings of plants. So there are various reasons for it. There are some biological reasons like fungi, bacteria, virus, phytoplasma, spiroplasma, nematodes, and many more. There are some non-infectious agents, like there could be nutrient deficiency which can affect plant health. There can be various abiotic stresses like drought, salt, frost, pesticides, and so on. Diseases caused by various infectious agents or bacteria, the agents, they are very, very harmful, and they spread very fast also. Among the infectious agents, fungi is the major organism. Plant diseases are the impairment of the normal function. This photograph I have taken from Agrios, which is a very famous book for plant pathologists. On the right hand side of the plant, there are diseases like root rot at the bottom, then wilting in leaves, leaf spots, fruit rots, leaf blights, shoot blight, crown gall. And on the other hand side, left hand side, the plant is very healthy. So this picture depicts how diseases can affect plant from tip to toe, from apical shoots to the terminal roots. Every part of plants can be affected by diseases. There can be cankers on stem, there can be crown gall on stem, there can be root rot, there can be flower rot, fruit rot, blight of leaves. I suggest you to go through another important article, The History of Plant Pathology in India. This describes at length how the science of plant pathology was developed in India. So as I told you, distribution of symptoms are from tip to toe. From top of the plant to the bottom of the plants, various diseases are there. And among these diseases, fungal diseases are the most important diseases. They are rated most important or sometimes second most important factor